Okay, so here is the next approach. So this is week for week number 2, and this pertains to the following files: endocrine pancreas and insulin action. Sorry, this is week 3, and these are the two lectures for this week. So the approach to try this time is to look at the figures and base all of the notes around that. So let's see, word of the day, medical terminology. I don't know what that is. Here we go, Dorland's online. So, word of the day, respirovirus, a genus of viruses of this specific family, respiratory infections. Cool. That's pretty straightforward. Okay, so moving forward with the figures, this is for the insulin action. What's that? Yeah, no, endocrine pancreas. So figure 1 is just this image of insulin in its precursor forms. Pretty straightforward. You can see things that get clipped off. So figure 2 out of 5, structural components of pancreatic B cells. So this is an individual cell isolated in order to illustrate what's going on. It looks like over here, this is the basal domain. So basolateral domain right here, this U shape, and up top is the apical domain. So we've got your nucleus, ER, Golgi apparatus, okay, secretory. We got the secretory pathway being illustrated. Cool, pretty straightforward. Moving on to figure 3. So this is pancreatic and intestinal processing of pre-pro glucagon. So there is these concepts of pre and then pro and then the actual molecule. So there's those 3 stages. So the before it's ready, and then the before it's ready, and then when it is ready. So in this case, it's glucagon instead of insulin. And let's look up real quick, what is glucagon? What is glucagon? Glucagon, it's a hormone which can treat severe blood sugar. It's a hormone. Okay. What is glucagon? So basic knowledge is simply that it is a hormone. We'll leave it at that for now. Next, we have figure 4 out of 5, and it says that the control of insulin synthesis and secretion. Oh, insulin, so how to synthesize it and how to secrete it. So AC stands for adenylyl cyclase, and mitose is mitochondria. So what do we have here? So on the right, it looks like this is maybe the outside of the cell. It's not labeled clearly, but this is probably extracellular space. And then glucose comes in, and maybe that's a transporter. I don't know. We have metabolism taking place, and then the products go to mitochondria. Mitochondria makes calcium, and then there's immediate secretion, protein kinase. Okay, so where does insulin come in? So, okay, so it looks like there is, the pathway through mitochondria is not direct. First, there is this swing around towards protein kinase, and then down towards insulin biosynthesis, and processing happens downstream after protein kinase has been activated. And also there's, through this other indirect path, there's another kinase involved, CAM, that might be cyclic AMP. We'll confirm with the notes. And that's also an indirect path to this end goal right here, to get to this insulin processing. And then the third indirect path is through something called AC, here it is, adenylyl cyclase, and then it activates cyclic AMP. This is definitely cyclic AMP, no doubt about it. And then protein kinase A. Cool. So we'll see how that relates to everything. And then here is figure 5 out of 5. So the role of the nervous system. It looks like we start here with the autonomous nervous system, and then parasympathetic. Okay, and then this thing, this might be an individual neurotransmitter, acetylcholine muscarinic agonist. So it increases insulin secretion. Okay. Okay, so this is on the left, you have an increase. Let's go ahead and label this. Let's go ahead and label that. It's pretty important. So an increase. We'll highlight that in green. You have your increase. And then a decrease over here. In red. Cool. So autonomic, and then sympathetic, parasympathetic. This I'll definitely have to redraw just to line it up better. But anyway, for now, the para. So para means stop, rest and digest. So you stop, rest and digest, and that's an increase in insulin secretion. That makes sense because you're digesting and then you want to have more insulin in order to perform the glucose uptake. As opposed to sympathetic, the run, what's it called? Fight or flight. You definitely need to decrease insulin in order to perform those functions. Cool. So we'll get around to that. So now we have the next topic, which is this insulin action. Figure 1 is the insulin receptor. Okay, cool. That's pretty nice. So on the top is outside of the cell, and then here is inside of the cell, and all of this I'll put together in one cell model to better make sense of it. So here, the insulin is binding to the alpha chains, and they transmit a signal through the transmembrane domain of the beta chains to activate the tyrosine kinase activity. Tyrosine kinase domain. Here we go. Very nice. So then this is figure 2 out of 5. You have again the extracellular space on the top and the cytoplasm on the bottom. So this is labeled as follows: activation of the tyrosine kinase domains of the insulin receptor by insulin binding, followed by interchain autophosphorylation. Cool. So that's an important term right there, autophosphorylation. So the insulin binds, and then something called IRTKL gets activated, and then something called IRTK but R gets activated, and then IRTKL again, it experiences phosphorylation. Cool. So these are the symbols of what each one means, what the symbols mean, and that one also I'm eager to redraw it. Then here's an overview of insulin action. Again, extracellular space is up top, cytoplasm is on the bottom. So you have glucose entering, good, and then insulin activator receptor tyrosine kinase. So this is the interaction between the receptor and glucose once it has been brought in, once it's present in the cytoplasm. So the interaction takes place. I'm not sure what it is. It just says metabolic responses for now. Anyway, there's the activation of protein phosphatase, which leads to dephosphorylation of a variety of enzymes involved in metabolism. Co-activation of protein. So the keyword here would be protein phosphatase. Yeah, we'll go ahead and label this, highlight this because that seems pretty important. We'll label it with this blue. So activation of protein phosphatase. Cool. Moving on to figure 4 out of 5, there is extracellular space again on top and then cytoplasm on the bottom. Looks good. Again, we seem to have an insulin receptor up top, and the caption says the following: the mechanism for mobilization of GLUT4 glucose transporter protein to the plasma membrane in muscle and adipose tissue in response to insulin. These are the abbreviations. Okay, cool. We'll get around to that. But let's see, just to get an overview of what's happening, there is a movement from this IRS thing down to the same IRS thing, but now it's phosphorylated, it looks like. So that attachment, yeah, the P probably stands for phosphate. So now it's active, the IRS is active, and then that interacts with this P85, probably a protein, and then kinase 3, and interacts with PIP2, and then it leads to the following. It leads to PDK, and then there's an activation. Phosphorylation. It may activate or deactivate this thing called AKT, and then AKT goes to the Golgi. It has something to do with the Golgi, and then GLUT4. So GLUT4 is involved there, and then GLUT4 is involved in other places. So I'm looking forward to learning what that is. Finally, figure 5 out of 5, you have this mechanism by which insulin promotes the cell growth and DNA synthesis via the RAS-MAP kinase pathway. The abbreviations used are IRTK, insulin receptor tyrant kinase. Cool. So now that goes back to one of the previous figures. We know what that. Well, now we know what this stands for, and this is a mechanism for cell growth. So this just focuses on cell growth. So we'll go ahead and label that with this yellow. Cell growth. Cell growth. There you go. Very nice. So that's the end of that.